Hello everyone, my name is Arutha Kulasinghe. I'm from the University of Queensland and we're at SITC in Houston today. I'll be telling you about a really exciting collaboration with Navinci that we've looked at in head and neck cancer. So I'll walk you through the poster. This was looking at a cohort of 32 head and neck cancer patients, pre-treatment patient samples, where we had the tissues prior to therapy and these patients were all given immunotherapy. And then we had the response data to checkpoint inhibitor therapy. So whether the patients had a pathological complete response, stable disease, partial response, or progressive disease. And so we used a combination of approaches of multiplex IF, combining the PhenoCycler fusion and the PSP technology with the Navinci PD-1, PD-L1 proximity ligation assay, so we could detect the interactions, not just the protein expression levels, but the actual interactions. A lot of these studies infer that, but what we wanted to do in this study was to really understand when you've got the receptor and ligand in close proximity, can we actually see that interaction? And using this technology, we can visualize that by rolling circle amplification that we can now visualize on the tissue. So this is a figure showing you PD-1 protein expression, PD-L1 protein expression, and the interaction PD-1, PD-L1 on the tissue directly. And so you can see that the interacting cells are indeed PD-1, PD-L1 positive, and the interactions are there in the tissue itself. And so here we're contrasting the different types of immune cells within the microenvironment at a composition level across those entire tissues. So I'll go to the really exciting bit of the microenvironment where we're mapping these across the whole tissues. So these are about 3 to 5 million cells that we've mapped here. So in the first sample, this is in a patient that had a pathological complete response at the top. So we've got the cell typing map, we've got the cellular neighborhoods, with the proximity ligation for PD-1, PD-L1, and then we've got the ISPLA signal itself. So the interaction is shown in the pink here, and this is the different levels going across here. So we've got the complete response, the partial response, the stable disease, and the progressive disease, and we've done this at a cohort level of 32 patients. And so what's really interesting is that we can see in the patient that had a complete response to checkpoint therapy, the interactions are really localized to these B cell zones within the tumor microenvironment, these TLS-like zones. And then when we contrast it to the non-responders, we can see that these indeed have PD-1, PD-L1 interactions, but they're almost restricted to the periphery of the tumor. And so this is where you've got the responder, the non-responder, and this is sort of your mixed signal that we're picking up in between that. So that's really interesting. So in a partial response, that's your staining pattern, and then in a stable disease. So these are sort of your intermediate responses to therapy. And so we can see really clear differences in the areas that are interacting in the responder versus the non-responder. And here what we've done, which is a more sort of complex analysis, is looking at the composition of having different cell types next to each other and how those are defined in the complete responder and the progressive disease patient. And so we've got a preprint on this work that's coming out in the next few days. Please look out for it. It'll be out on Research Square and hopefully in a publication in the near future. Thank you.